So this is just a really short video. It's going to go over the scientific method with you, and it's just going to remind you what the scientific method is, and I want to just touch on how we're going to be applying it in the lab. So I actually really like this diagram of the scientific method. I didn't show it to you in grade 9 because it was a little intense, but I think that now that you're in grade 11, you can appreciate it a little bit more. Let's zoom out so you can see. And here you can actually see the scientific method is actually really circular. So there's really necessarily no start or no end because you're constantly doing it. So there's really no finish to the scientific method if you're doing it correctly. So we're going to kind of start here. And usually in the scientific method, you have to make observations. So you have to make some sort of observation. Now, this always kind of scared me in university because I always thought they had to be novel observations. They don't have to be novel observations. It doesn't have to be something that no one else has studied before. Some of the best discoveries that we've had in science have been based off of someone trying to replicate someone's research. So you're just going to look around you and you are going to make observations either that you can see. Typically in chemistry, it's things that you can see based off of matter. But there's other branches of science that it could be based off of feelings or how things behave or nature. Once you have an observation, you're going to think of some sort of interesting question. So maybe you're going to notice a pattern. Maybe you're going to ask why something changes color. Maybe you're going to ask why something changes shape. So you're going to have that interesting question. You're then going to formulate that question into a hypothesis. Why do you want to put it into a hypothesis? Because a hypothesis is much easier to test. So if you're just asking, why is the sky blue? That's really not very easy to test. But if you can go through and make some sort of prediction, if-then statement, it's like your best guess of why something is happening, that's much easier to test. So for example, let's go back. For those of you that have me in grade 9, you had that grass lab that you had with me. So let's go back to that. I think that grass will grow taller when more water is added. So rather than asking the question, will grass grow with water? You're going to go ahead and you're going to use an if-then statement to actually state what you think is going to happen. Obviously, you're not going to use I in a hypothesis. So your hypothesis would be, if water is added, grass will grow taller. Once you actually have your hypothesis, you can actually go through and develop testable predictions. And once you have your testable predictions, you can actually develop a procedure. So you would go through and develop an actual procedure on how to test your hypothesis. How would you test if adding water is going to make grass grow? Once you actually develop a procedure, you're going to go ahead and you are going to gather data. This is what's known as doing the experiment. So you're actually going to gather data and you're going to go through and you're going to undergo your experiment. So you're going to do your experiment to gather data to either accept or reject your hypothesis. Once you have your data, you're going to go back and you are going to refine your hypothesis. So odds are is that the first time that you actually do this, the first time you go through, your procedure is probably not going to be very good, and that is not your fault. So typically, it's really hard to actually go through and develop a really, really, really good experiment that is going to test something while limiting other variables. So you might go to actually do the experiment and realize that you weren't controlling this variable. So you got to go back to the drawing board. And then you might go through and then all of a sudden your experiment doesn't work. You don't know why. So you got to go back to the drawing board. So typically you're going to spend a really long time in this little loop here. Not only because you might have problems with your procedure, once you actually start nailing your procedure, maybe then you'll see that your data is actually not supporting your hypothesis. So you're going to want to go back and you're going to want to readjust your hypothesis. Maybe water doesn't make grass grow taller. Maybe too much water actually causes the grass to not grow at all. So you want to go through, you want to refine your hypothesis, and eventually you're going to get out of this loop because you're going to have a hypothesis that you actually have proven to be correct over and over and over and over again. Once you have that, then you can go ahead and develop a general theory. So all of those theories that we actually are going to be studying in chemistry, any theory that I show you, starting with next week's lesson, atomic theory, they're all based off of this little loop here. So someone actually made a hypothesis, figured out an experiment, tested it, it went horribly wrong, so they had to go back to the drawing board over and over and over again. I like to talk about this to start because oftentimes you are going to have labs that don't work. Might be your fault, might not be your fault, right? Maybe you didn't read the instructions properly and your lab data actually didn't support what you should have seen. Maybe I made the concentrations wrong. Maybe there was some other variable that you didn't even know about that was impacting it. So the downside of this class is we typically only do a lab once. So we're going to have a hypothesis, we're going to have our procedure, and we're only going to do a lab once and try to make conclusions. That's really not the real scientific method. So I want you to actually be really happy when you make a mistake on a lab, because when you make a mistake on a lab, it's actually like real science. It's actually going through this loop again, and really we learn from our mistakes, which is, again, something that I'm going to try to touch on when we talk about atomic theory next lesson. So I'm going to keep touching upon all the mistakes that were made and how we actually discovered stuff. I just got a package. That's exciting. Now my dog's going to bark. I'm sorry. So based off of this, very exciting. I wonder what it is. Based off of this, there's also two types of observations that we're going to be making in a lab. So the two types of observations that we're going to be making in a lab are called, the first one is called qualitative. So a qualitative observation is based off of non-numerical observations. So it's when there's no numbers that can be taken. So for example, me describing how red something is, there's not really numbers that I can apply to that. So things like color, things like state, things like shape, when you're describing those things to me, you are describing qualitative observations. In chemistry, there's a second type of observation, and that is called a quantitative observation. We really like quantitative observations in chemistry. So qualitative observations are really easy, but if you can make a quantitative observation, please, please, please do so. So a quantitative observation is based on numerical observations. So you can actually apply a number to your observation, so it's more specific. So for example, if something has a mass, make sure you tell me the mass, because that's an exact number. Length, volume, those are all quantitative observations. So we're going to make as many of these as possible in chemistry. Hello, boy. We're going to make as many of these as possible in chemistry, and then we're going to fill in the gaps with qualitative observations. Last thing I want to talk about with making good observations. Good observations should be objective. So what objective means is, okay, it's fine. What objective means that you are not biased at all. So you don't want to go in trying to prove your hypothesis. You just want to go through and you want to make an experiment and make observations. So all of your observations should be objective. So don't go in looking for something, just record what you see. Again, some of the best observations that we've made have been based on mistakes. You want them to be as accurate as possible. So again, use numbers as much as you can. If you can't use numbers, use descriptive words. Like use words like dark red or light red. Don't just tell me red, because that really doesn't tell me anything. Observations should also be concise though. When I look at an observation table, it should be just that, a table. I shouldn't have to read a paragraph in your observation table. You should have descriptive words, but you want it to be as concise as possible, not with any unnecessary words. Last thing I want to talk about is this thing that you might be tempted to throw out, but definitely don't throw out. And it's called an outlier. Sometimes you can hear it called an outlier as well too. An outlier is just an irregular result. So it's something that unexpected happened. Let me rephrase that. It's something unexpected that happened. So it's something that you didn't think was going to happen that did. Sometimes we call this an anomaly as well too. Even though it doesn't fit the pattern, because we're going to be making some graphs and stuff like that too. Even though an anomaly or an outlier might not fit the pattern, doesn't mean it's actually bad information. A lot of the time, some of our best discoveries have been made based off of anomalies. So usually there's a logical explanation for why that anomaly is happening. You just might not understand it yet. Maybe it's something that we cover in grade 12 chemistry. Maybe it's something we cover in university chemistry. And maybe you've just made a really cool new discovery that you're going to study later on down the road. So anytime you have an unexpected lab data point, don't throw it out. Record it. You can discuss it in the conclusion. So anytime you make a mistake, I say you can always discuss it in the conclusion, and maybe you've made some sort of cool discovery. So this is going to be how we are going to be doing science this semester. This is going to be how we're doing labs this semester. I do have a pretty specific lab format that I want to follow that I'll be touching on later on. Next up, we are going to be talking about the atomic theory. So again, theories, scientific method, and everything we know about atoms. So I'll see you in the next video when we talk about atomic theory.